Delivering pharmaceutical compounds to a specific cellular target requires crossing a restrictive physical barrier, the cell membrane. Most drugs cannot pass through this bilayer alone. To solve this permeability problem, researchers in a 2018 study synthesized a series of novel molecules called lactose-based fatty acid monoesters. These synthetic molecules were designed to act as chemical escorts, increasing the permeability of mucosal barriers so a therapeutic payload could reach target cells. During testing, however, researchers recorded high levels of cytotoxicity. At specific concentrations, these synthetic escorts killed the cells they were engineered to treat. To determine why this delivery vehicle becomes lethal, we must isolate the point of cellular failure. In this case, the breakdown occurs within the mitochondria. The researchers measured mitochondrial health using a fluorescent assay known as the J aggregate to monomer ratio. A high ratio indicates the mitochondrion is holding a negative charge across its membrane. A drop in this ratio signifies that the membrane potential has dissipated, preventing the mitochondrion from synthesizing ATP. This chart tracks mitochondrial health. The y-axis is the J aggregate ratio. The x-axis is surfactant concentration. Lactose caprate, a 10-carbon chain, requires 1 millimolar to drop the potential. As researchers tested 12, 14, and 16 carbons, the curve shifted aggressively leftward. The 16-carbon variant, lactose palmitate, reaches maximum cytotoxicity at a fraction of the dose required for the 10-carbon molecule. The addition of 6 carbon atoms to the tail of this molecule shifts the concentration required for mitochondrial death by several orders of magnitude. The specific length of the hydrocarbon chain dictates the survival of the cell. To explain that shift, we must look at the thermodynamic rules governing biological lipids, starting with the concept of the amphipathic molecule. A phospholipid has a polar head attached to a nonpolar tail. Water bonds with the head but repels the tail. To minimize repulsion, lipids form a micelle, bearing tails inside. At cellular concentrations, this pressure forces lipids into expansive sheets, creating the phospholipid bilayer. Cell membranes are dynamic, fluid structures held together entirely by the continuous thermodynamic repulsion between water and carbon. Because the membrane is held together by physical forces, its structural integrity is dictated by the geometry of the fatty acid tails. A saturated fatty acid contains only single bonds. This straight chain packs tightly against neighbors, maximizing van der Waals forces. An unsaturated fatty acid contains double bonds. This feature creates a rigid bend, physically pushing neighboring tails away and increasing space between them. This molecular geometry scales to macrophysical states. Saturated fats pack tightly to form solids like butter, while unsaturated fats remain fluid liquids like olive oil. These tails are anchored by a structural backbone. Glycerophospholipids use a 3-carbon glycerol backbone, while sphingolipids are built on a sphingosine chain. While the tail controls the physical state, the polar head group dictates the cell's biological identity. The ABO blood typing system is based on the specific sugar moieties attached to the heads of red blood cell sphingolipids. The hydrophobic tail controls the structural physics of the membrane, while the hydrophilic head controls the biological function of the cell. The body uses a different class of lipids for energy storage, triacylglycerols. These molecules consist of 3 fatty acid chains bonded to a glycerol backbone via ester linkages. Exposing these energy stores to a strong base, like sodium hydroxide, triggers a cleavage reaction known as saponification. The sodium hydroxide cleaves the ester bonds. This yields free fatty acid salts, or soap. This molecule is a surfactant, capable of lowering the surface tension between 2 distinct phases. Surfactants form micelles to sequester hydrophobic particles inside a nonpolar core, while the exterior polar heads allow the entire package to dissolve in water. Any molecule engineered with a distinct polar head and a nonpolar tail will act as a surfactant, attempting to organize itself to minimize thermodynamic tension within a lipid environment. We can now return to the 2018 study of lactose-based fatty acid monoesters. The researchers utilized a bulky lactose sugar moiety, which acts as the hydrophilic polar head. Attached via an ester bond is a saturated hydrocarbon chain, serving as the hydrophobic nonpolar tail. Although synthesized for drug delivery, the physical structure of this molecule matches a biological surfactant. When introduced to a cell, Thermodynamic properties force the molecule to seek out and embed into existing lipid structures. The drug's amphipathic geometry compels it to integrate into the cell's architecture, moving beyond the intended role of a passive carrier. The mitochondrion is particularly vulnerable because it relies on an impermeable inner lipid bilayer to trap a dense concentration of protons. This gradient provides the energy required for ATP synthesis. As the synthetic lactose surfactants approach the inner mitochondrial membrane, they insert their hydrophobic tails into the bilayer to escape the surrounding water. A 10-carbon tail is too short to reach the membrane's core, leaving it intact. The 16-carbon tail matches the core's thickness, anchoring deep. The bulky lactose head acts as a wedge, forcing native lipids apart. Through this gap, stored protons leak, bypassing ATP synthase. This physical leakage is precisely what the J-aggregate ratio recorded. The plunging line was a real-time measurement of the mitochondria depolarizing as holes were wedged into their surface. Biology is applied chemistry. Mastery of lipid geometry, amphipathicity, saturation, and chain length reveals the mechanics of cellular assembly and the precise method of its destruction.